My name is Tim Corson. I'm Professor and Chair of the Department of Pharmacology and Toxicology at Indiana University School of Medicine in Indianapolis, Indiana, USA.
Please tell us about your presentation here at ARVO this year.
Here at ARVO, we presented some exciting research from my lab focused on a new delivery system of an agent that we think has potential for treatment of the wet or neovascular form of age-related macular degeneration. My lab has been interested for a number of years in trying to find new potential therapeutic targets that might be separate from the commonly used VEGF targeting agents. We identified an enzyme in the cell called ferrochelatase that's involved in making heme. Heme is the red pigment that gives blood its color, but it's important for all kinds of proteins in the cell. And ferrochelatase catalyzes the very last step in the formation of heme. We've shown that ferrochelatase is important for angiogenesis, the process of blood vessel growth, in a variety of different systems, and is also highly expressed in diseases like wet AMD. There's an old drug called griseofulvin that's been around since the 1950s that is used to treat fungal infections, but it also has what we call an off-target effect of inhibiting ferrochelatase. So we think we can repurpose griseofulvin to inhibit ferrochelatase and therefore to treat wet AMD. The challenge we face, of course, is making sure that we can deliver griseofulvin to the right place and with a duration of effect that would allow some therapeutic use in the disease. So in the project we presented here at ARVO, we worked with a formulation scientist, Professor Yoon Yo at Purdue University, to create a new formulation of griseofulvin that is able to release the drug over a long period of time in the eye. So we created what are called microparticles, which are tiny little beads that can degrade in biological fluids, embedded with griseofulvin so they can release the griseofulvin drug over a period of more than 30 days. In the work we presented here, we showed that these particles could release drug that was able to stop the growth of blood vessel cells in culture, and also in an animal model of choroidal neovascularization, which is the type of blood vessel growth that occurs abnormally in wet AMD. So we think these particles, with some further optimization, have real potential for a therapy that could be injected like the existing anti-VEGF drugs, but have a nice sustained effect over weeks or even months that could be combined with the existing drugs or perhaps even be a standalone therapy in future.
So this is another injection. Are you considering other delivery methods?
Yes, so this is an injectable therapy. It might be possible to deliver griseofulvin systemically, so it is taken as a pill for fungal infections, but to be sure of getting high concentrations within the eye without the risk of side effects, we thought it was preferable to try and deliver this kind of sustained release formulation directly to the eye. And of course, although intravitreal injections are not something that anybody really enjoys, it is the standard delivery route for the existing drugs. So if we were considering a potential combination therapy in future, we could use the same injection technique that is currently used for anti-VEGF agents.
Does this drug seem to work similarly to other anti-VEGF drugs by attacking blood vessel growth?
So the mechanism of griseofulvin, by inhibiting this enzyme ferrochelatase, it shuts down the production of certain proteins that rely on heme in the cell, and some of these proteins are important for the growth of blood vessels. That is the underlying pathology in the wet form of AMD. So by blocking heme synthesis, we're able to block the function of certain proteins that are important for blood vessel cells to divide. However, we have shown that griseofulvin and ferrochelatase inhibition more broadly is not toxic to the existing blood vessels, but really stops the new ones from forming, which of course is exactly what you would want in the context of a therapy that is safe for the normal blood vessels in the eye, but stops the formation of the new neovessels that occurs in wet AMD.
You've completed this project in cell models and through animal models. What are your next steps?
So we'd like to optimize our microparticles further as a next step, perhaps to increase release time even further, out to the level of months to be competitive with some of the newer therapies that are now coming onto the market. Everything we're using in these microparticles is considered safe for drug use, so we think it could be quite a rapid path towards development of these particles for human use. But we do still have to do quite a bit of validation and, of course, safety studies before we're ready to launch any kind of clinical trial.
Would you like to highlight any other studies you have ongoing in your lab?
Yes, in my lab we have a variety of projects under this big umbrella of trying to find new targets and potential new therapies to block abnormal blood vessel growth as seen in wet AMD. And we also have a little interest in dry AMD, but primarily we're focused on anti-angiogenic proteins and then small molecules that target them. One other area of research that we're very excited about is a protein called Ref-1, which is involved in regulating transcription factors in the cell. So these are the cellular components that control which genes are turned on and off at different times. And Ref-1 blocks the function of a variety of transcription factors that are important for the growth of cells, for inflammation, and for angiogenesis. So we think that if we can block Ref-1, we can shut down a variety of pathways in the cells that are hyperactive in the context of wet AMD, including especially inflammation and angiogenesis. Ref-1 is a very exciting target because there is already a drug that has been tested in humans called APX3330, APX3330, that was initially tested in the cancer context and shown to be well tolerated in patients, and recently completed a phase 2B trial in diabetic retinopathy and diabetic macular edema, where it did show some preservation of visual function in those patients. So in my laboratory, we're working on some of the biological validation of Ref-1 as a target, exploring how we can best inhibit it in models of wet AMD. But with the clinical data already out there in other indications, we're very excited about the possibility of rapidly transitioning that APX3330 drug into a new therapy for wet AMD. And the exciting part of that drug is that it is an oral drug, so it could be taken as a pill twice daily, avoiding the need for intravitreal injections.
Are either of these potential treatments going to clinical trials anytime soon?
So neither our griseofulvin story or the APX3330 story are quite ready for clinical trials for wet AMD. However, since griseofulvin is already an approved drug, we think it could progress quite rapidly in that direction. And APX3330, of course, in its current form, is already being used in clinical trials for diabetic retinopathy and diabetic macular edema, so it could be quite rapidly translated to new clinical trials for wet AMD. We're not quite there yet, but we're pretty close.
Is there a website people can follow this research and possibly get involved in any upcoming trials? Is there anything you'd like to close with?
I'd like to close by thanking the funding sources for the research that we do, primarily the National Eye Institute, part of the National Institutes of Health, and the griseofulvin project began with a grant from the Bright Focus Foundation. So we're very thankful for the research support provided by these government agencies and foundations that help us to develop what we hope will be exciting new therapies for AMD in future.
Thank you so much for sitting down with us, and we're excited to keep up with your progress.